Cancer is scary. Around 1 in 6 deaths globally are due to cancer, and many cases are untreatable. Why doesn't a disease like this trigger an immune response, and what can we do about it? Let's start with your immune system. Antigens are little identifiers on the surface of cells. Our immune system has white blood cells, namely T cells, that use antigen receptors to latch onto these identifiers and destroy the cell that has them, usually a foreign invader. Cancer is often undetectable by your T cells because cancer cells have similar antigens when compared to your normal cells. If there is a blood cancer like leukemia, the treatment is even more difficult because the cancer is moving around in the blood. Often, surgery to remove this type of cancer is impossible. What if your immune system could identify and terminate these cancer cells in your blood? This is where the chimeric antigen receptors, or CAR, comes in. CAR T cells have a modified antigen receptor that is able to identify and latch onto cancer cells that have a matching antigen. These modified antigen receptors are created by modifying the genetic code of the T cell itself. First, a sample of blood is taken from the affected patient to examine and modify the DNA of their T cells. To modify the genes of the patient's T cells, we have to start the process of DNA printing. Small chains of nucleotides called oligonucleotides or oligos are printed in the lab to form the genes a patient needs. First, a container with an array of tiny electrodes is submerged in a solution that contains one of the 4 types of nitrogenous bases. When a certain electrode is activated, the ends of the nucleotides are no longer protected, which then adds a single nucleotide to the tip of the phosphoramidite on that electrode. Scientists can then vary the charge of the electrode and change which nitrogenous base is present to build oligos from the ground up, nucleotide by nucleotide. Once the genes are printed, we move on to the insertion of these genes into the patient's T cells using CRISPR. CRISPR is like an immune system on the atomic level that is used by bacteria. CRISPR is a section of DNA that is made up of repeating patterns of the nitrogenous bases. These repeats are separated by spacer DNA. Each spacer DNA section is a portion of a foreign invader's genome. Whenever there is a new invader in the cell, a protein attempts to take it apart. A small part of that foreign DNA is then added to a space between 2 repeating segments, like a library of past infections. There are also CRISPR-associated genes, or Cas genes, that are responsible for the protein that move all this DNA around. When an invader infects a cell, a different protein complex is made that takes an RNA transcription called CRISPR RNA from spacer DNA and compares the transcription with the foreign DNA. If the sequence matches, the foreign DNA is cleaved, rendering it harmless. Essentially, it's responsible for cataloging past invaders and defending the bacterium from foreign DNA. Scientists have found a way to hijack the system and use it to modify the very DNA of a patient's T cells. There is a specific protein complex called Cas9 made in the bacteria S. Pyogenes. Cas9 identifies foreign DNA and cleaves it if it matches with the CRISPR RNA. If we insert our own strand of guide RNA and feed in the patient's T cell DNA, the DNA can then be cut wherever we want. Once the DNA is cut, we can insert our synthetic genes. These genes will be used by the CAR T cell to make specialized antigen receptors to fight the cancer. The patient is given a dose of chemotherapy to reduce the number of other immune cells, giving the CAR T cells a better chance to fight the cancer. Then, the patient's blood is infused with the CAR T cells, and they get to work destroying the cancer. Providing an alternative to chemotherapy, as well as sustaining a 94% chance of remission, CAR T cell therapy can be a great help with our current problem with cancer, using modern technology to defend from what our immune systems can't see. From the printing of DNA to synthesis of CAR T cells, there's an incredible amount of research that goes into this treatment. Although this technology is still in its infancy, there is great potential for the future of medicine. Thanks for watching.